All of you can hear me? Hello, good morning.
Morning, ma'am. Yes, ma'am.
Okay. I think there is no any doubt about the iron metabolism, about what is the transferrin concentration, what is TIBC, what is transferrin saturation. Everything is clear. What is TIBC? Roll number 4. Who is roll number 4? Roll number 4 is there. What is TIBC? If you know, just tell me. No, if you don't know, you just tell me. No, you don't know, like that. No. Roll number 6. Can you hear me? All of you can hear me?
Yes, ma'am.
Yes. Anybody knows? You can tell me. Just you see the in the books also. You can tell me. What is TIBC? Ultimately, you have to know, because if you will come, if you will be asked in, it will be asked in your exam. No, then you have to understand. Okay. Transferrin concentration, then TIBC and transferrin saturation. It is not that easy, isn't it? Anybody can tell me what is TIBC? What is the difference between the TIBC and transferrin saturation? That means you didn't understand. This iron panel already finished. Okay, this iron panel will be asked in your exam. Transferrin concentration. Even though transferrin concentration is 300 to 350 milligram per dl, TIBC, that much of transferrin can bind only 300 microliter, 300 microgram per dl transferrin. Iron. Okay, even though transferrin concentration is 300 to 350 milligram per dl, okay, that much of transferrin is sufficient for binding only 300 microliter per dl, 300 microgram per dl, okay, that much of iron. Okay, and at that time all the binding site, even though transferrin can bind, okay, one transferrin molecule can bind two atom of iron, ferric iron. Okay, but at the same time all the binding sites of the transferrin are not occupied. At the same time all the binding site of the transferrin are not bound by the iron. Okay, that's why at the same time transferrin is not fully saturated. Okay, normal transferrin saturation is 30 to 35, okay, percentage. Okay, 30 to 35 percentage, isn't it? All the binding site of the transferrin is not occupied by the iron at the same time, isn't it? Only 30 to 30% is occupied. Even though total transferrin concentration, normal total transferrin concentration is 300 to 350 milligram per dl, okay, that much of transferrin is sufficient to bind 300 microgram per dl iron. Okay, that much of capacity, they can buy, that transferrin can bind 300 microgram per dl iron, but at the same time all the binding sites are not saturated. Okay, that's why TIBC is different and transferrin saturation is different. Okay. The iron concentration, whatever iron concentration in our body, that is the saturated iron binding capacity, isn't it? Our body iron concentration is the saturated iron binding capacity of the transferrin, isn't it? Saturated iron binding capacity of the transferrin, because anyhow the iron is going to be transferred via transferrin. Okay, so unsaturated iron binding capacity can be calculated by TIBC, total iron binding capacity, minus body total iron. Body total iron means saturated iron. That iron is bind with the transferrin, isn't it? So that's why that saturated iron, that means the total body iron divided by total iron binding capacity. How much available binding site in the transferrin? Okay, how much iron maximum they can bind, that is TIBC, isn't it? TIBC. Okay, so present iron concentration, total iron concentration divided by TIBC into 100. This figure will give you, this result will give you transferrin saturation. Okay, transferrin saturation. That means out of this much of TIBC, our body saturated iron is this much. So if it is 100 microgram, so how much our that body iron concentration, that is the transferrin saturation. Okay, transfer in saturation. Now I'll going to start this calcium metabolism. So calcium, the total calcium in the human body is about 1 to 1.5 kg, 99% of which is seen in bone and 1% in extracellular fluid. Sources of calcium. Milk is a good source of calcium. Calcium contents of cow's milk about 100 mg per 100 ml, okay, or 100 mg per deciliter. Egg, fish and vegetables are medium sources for calcium. Cereal, that is wheat and rice mostly, contains small amount of calcium. But cereals are staple diet in India. Therefore, cereal are the major source of calcium in the Indian diet. Daily requirement of calcium. An adult needs 500 mg per day and a child about 1200 mg per day. Okay, calcium. The requirement may be increased to 1500 mg per day during pregnancy and lactation. After the age of 50, there is a general tendency for osteoporosis, which may be prevented by increased calcium, that is up to 1500 mg per day and plus vitamin D, okay, 20 microgram per day. Mechanism of absorption of calcium. Okay, mechanism of absorption of calcium. Absorption is taking place from the first and second part of duodenum. Calcium is absorbed against a concentration gradient, and this is a energy requiring process. Okay, because it transport against the concentration gradient, okay, by the active transport, isn't it? So it must be required energy. Absorption requires a carrier protein helped by calcium dependent ATPase. Out of 500 mg calcium taken orally per day, 400 mg is excreted in stool and 100 mg is excreted through urine. Okay, out of 500 mg calcium taken orally per day, 400 mg excreted, in major part is excreted via stool and 100 grams is excreted via urine. Factor causing increased calcium absorption. Okay, vitamin D. Vitamin D is very much essential for calcium absorption. Okay, calcitriol induces synthesis of a carrier protein. Active form of the vitamin D, calcitriol. Okay, calcitriol 125 dihydroxy cholecalciferol. Okay, that is called calcitriol. Induce synthesis of carrier protein calbindin in the intestinal epithelial cells, and so this facilitates the absorption of calcium. Parathyroid hormone increases the calcium transport from the intestinal cell. Acidity favors the calcium absorption. Remember, acidity, we need the media acidic, okay, for calcium absorption. So acidity favors the calcium absorption. Amino acid like lysine, arginine increase the calcium absorption. Factor causing decreased calcium absorption. Those are phytic acid. Okay, phytic acid. Hexaphosphate of inositol is present in cereal. Phytic acid. Okay, it's decreased calcium absorption. Oxalate present in green leafy vegetables, which causes formation of the insoluble calcium oxalate. Okay, it forms the calcium oxalate which is very much insoluble. And in case of malabsorption syndrome, here the fatty acid is not absorbed, okay, causing formation of insoluble calcium salt of fatty acid. Okay, in case of malabsorption syndrome, fatty acids are usually not absorbed. Okay, this leads to formation of the calcium salt with the fatty acid, isn't it? Fatty acid. Phosphate, high phosphate content will cause precipitation as calcium phosphate. The optimal ratio of calcium to phosphorus, which allows maximum absorption, is 1:2 to 2:1. Okay, that is the optimal ratio of calcium to phosphorus, which allow the maximum absorption is 1:2 to 2:1. Okay, the milk contain this optimum ratio. Milk contain this optimum ratio. This is how calcium is absorbed from the intestinal lumen. Okay, in the intestinal lumen, dietary calcium, okay, they will form this one, calcitriol receptor complex, okay. This calcitriol induce the synthesis of one protein, calcium binding protein, calbindin, isn't it? Calbindin, okay. That calbindin binds with the calcium and calcium is get absorbed, okay. This CR for calcitriol receptor, complex, called calcitriol receptor complex. Actually, this calcitriol is the active form of vitamin D. It induced the synthesis of the calbindin. Okay, this calcitriol induced the gene, okay, genetic sequence that is responsible for coding the protein calbindin, isn't it? Calbindin in its turn, it's bind to the calcium and calcium is get absorbed. Okay, this is how calcitriol or active form of vitamin D helps in calcium absorption. Okay, calcium in cells. Calcium is mainly extracellular. Okay, it is mainly extracellular. The cell membrane is generally impermeable to calcium ion. Okay, generally in the normal state, okay, the cell membrane is impermeable to calcium ion. Okay, so calcium influx into the cell, so we need some transporter, active transport, isn't it? Because extracellular concentrate, it is a mainly extracellular ion, okay, because the cell membrane is quite impermeable to calcium ion, so we need the transporter mechanism, okay, we need transporter. So calcium influx into the cell by sodium calcium, not sodium potassium, okay, sodium calcium exchange mechanism, okay, exchange mechanism. This mechanism is rapid but low affinity, okay, it is very fast but affinity is low affinity pump, okay, it is low affinity for calcium, okay, it will come as a short notes, okay. Sodium calcium exchange mechanism or sodium calcium ATPase, okay, exchange mechanism is a low affinity but rapid, okay, low affinity exchange mechanism. Entry of calcium into the mitochondria by calcium uniport system. Okay, intracellular, once it is internalized to the cell, it has to be transported to the mitochondria for certain activity, isn't it? For certain its function. So entry of the calcium into the mitochondria by calcium uniport mechanism. Okay, calcium uniport mechanism or calcium uniport system. But calcium exit by, okay, but calcium ion exit by sodium calcium antiport system. Okay, exit of the calcium by the sodium calcium antiport system, which in turn is dependent on sodium proton ATPase pump. Okay, sodium hydrogen plus, okay, that is sodium proton ATPase pump. This is called calcium pump. Okay, this is also called calcium pump. Other calcium channels are voltage operated channels and second messenger operated channel, SOC, second messenger operated channel. Okay, these are the voltage gated channel, okay, second messenger operated channel, okay, second messenger operated channel. This one is the hormone receptor operated channel. Here is the sodium calcium exchanger, okay, sodium and calcium exchanger, okay, calcium is internalized and sodium is externalized, isn't it? And the calcium uniport system is there. Once it is calcium is externalized, it has to be transported to the mitochondria by the calcium pump uniport system. Okay, uniport system, entry of the calcium into the mitochondria by calcium uniport system. This is the calcium uniport system. And exit of the calcium from the cell, it is depend on the sodium hydrogen sodium proton ATPase pump, isn't it? sodium proton ATPase pump or ATPase pump. It is depend on sodium proton ATPase pump, isn't it? So this exit by, no doubt this exit by sodium calcium antiport system, but this antiport system is dependent on sodium proton ATPase pump. Okay, this is the, you can say, the secondary active transport. Sodium calcium antiport system, it is in one sense you can call the secondary active transport. Okay, secondary active transport. Here what happened? This is the primary active transport, sodium proton ATPase pump. It causes externalization of the sodium, okay, against the concentration gradient, because extracellular sodium ion sodium is a extracellular ion, okay, extracellular major extracellular cation, okay, it is externalized, okay, against the concentration gradient, okay. At that time proton is internalized, okay, and this drive force the again, this drives makes the concentration of the sodium in the extracellular fluid, it is becomes more high, isn't it, more high. Once it is more high, secondarily towards the concentration gradient, sodium flow occur. That means sodium is internalized, okay. At that same time that calcium is externalized to maintain the electrochemical neutrality, okay. This is the main function of the sodium calcium antiport system, which is dependent on the sodium proton ATPase pump, okay. Now functions of calcium, functions of calcium functions of calcium, activation of enzymes, okay, and activations of the enzyme. Only ion, please remember, only ionized calcium is metabolically active, okay. Metabolically active form fraction of the calcium is ionized fraction, remember. Activation of the enzyme. Calmodulin can bind 4 calcium ion. Calmodulin is a protein, okay. Remember calmodulin, it is a protein, okay, which can bind 4 calcium ion, okay. Calcium binding leads to activation of the enzyme. Once calmodulin bind with the 4 calcium ions, this binding, okay, leads to activation of the enzyme. Because calmodulin, it is a part of various regulatory kinases, okay. In the metabolism part, you have gone through to the various kinases mechanism, okay, kinases, okay. So calmodulin is a part of various regulatory kinases, regulatory enzymes, okay, where the calmodulin, it is an integral part, isn't it? Some other enzymes are activated directly by calcium without the intervention of calmodulin. Some of the enzyme calcium can activate directly, okay, directly. Nerve conduction. Calcium is necessary for transmission of the nerve impulses from presynaptic to postsynaptic region. In the muscle also it has got some role. Calcium mediates the excitation contraction of the muscle fiber. Okay, in the excitation contraction coupling also calcium required. Okay, it plays a major role, okay, in the excitation and contraction of the muscle fiber. Upon getting the neural signal, calcium is released from the sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. That is, the sarcoplasmic reticulum is the endoplasmic, nothing but the endoplasmic reticulum of the muscle fiber. Okay, muscle cell. Okay, in the muscle cell, endoplasmic reticulum is known as sarcoplasmic reticulum. That calcium activate ATPase, increase the action of actin and myosin, and facilitates the excitation contraction coupling. The trigger of muscle contraction is interaction of calcium with troponin C. Calcium decreases the neuromuscular irritability. Okay, it decreases the neuromuscular irritability. Secretion of hormones. Calcium mediates the secretion of insulin, parathyroid hormone, calcitonin, vasopressin, okay, vasopressin, etc. from the cell. Okay, these are the hormone: insulin, parathyroid hormone, calcitonin, vasopressin. Okay, from the cells, and it can act as a second messenger also. Okay, calcium and cyclic AMP are the second messenger of different hormones. One example is glucagon. Okay, calcium is used as the second messenger in system involving the G protein and inositol triphosphate. Under the mechanism of action of the hormone, you will get how calcium is used as a second messenger in the system that is involved for G protein and inositol triphosphate. Okay, phosphoinositol triphosphate actually. Okay, these are acts as a second messenger mechanism. Without calcium, this operation of this second messenger mechanism is not possible. Okay, vascular permeability. Calcium decreases the passage of serum through capillaries. Thus, calcium is clinically used to reduce allergic exudates. Okay, allergic exudates, to reduce the allergic exudate as it decreases the passage of serum through the capillaries. Coagulation. Calcium is known, as everybody knows, it is a factor 4 in the blood coagulation cascade. Prothrombin contain prothrombin contain gamma carboxyglutamate residues, which are chelates by calcium during thrombin formation. Okay, prothrombin contain gamma carboxyglutamate residue. Okay, gamma carboxyglutamate residue which chelate by calcium during thrombin formation. Okay, gamma carboxyglutamate formation requires vitamin K, remember. Okay, from the glutamic acid it has got, okay, how many carboxyl group? Glutamic acid is what type of amino acid? Anybody? Glutamic acid, what type of amino acid is glutamic acid? Anybody can tell me? Huh?
Acidic.
What type of amino acid? Tell me.
Acidic.
Acidic amino acid. So it has two carboxyl group, isn't it? Two carboxyl group. One is alpha, another is what? Another is, one is alpha, another is gamma carboxyl. Okay, gamma carboxyl group. Okay, glutamic acid. So gamma carboxyglutamate. Okay, prothrombin contains gamma carboxyglutamate residue. Okay, gamma carboxyglutamate residue, which chelates the calcium during, okay, during the thrombin formation. Okay, so myocardium. Calcium prolongs the systole. Calcium prolongs the systole. Okay, in hypercalcemia, cardiac arrest is seen in systole. In fact, should be kept in the mind when the calcium is administered intravenously. Okay, it has to be very conscious. Okay, it should be given very slowly. Okay, during the IV infusion, you have to give very slow infusion. Okay, because calcium prolong the systole. Okay, bone and teeth. The bulk quantity of calcium is used for bone and teeth formation. Okay, bones also acts as a reservoir for calcium in the body. Osteoblasts reduce the bone absorption and osteoclast produce demineralization. Okay, osteoblasts induce bone deposition and osteoclasts produce demineralization. How glutamic acid is transferred to gamma carboxyglutamate? Okay, gamma carboxyglutamate, you have to study the vitamin K. Okay, vitamin K is responsible for transformation of glutamic acid to gamma carboxyglutamate. Okay, gamma carboxyglutamate. Okay, that residue in coagulation part. Okay, vitamin K is responsible for formation of glutamic acid to GLA. GLA means gamma carboxyglutamate. Okay, which chelate by calcium during thrombin formation. Okay, this is the mechanism of action of calmodulin. Once calcium bind with the calmodulin, okay, this bound calmodulin this active some protein kinases, so active kinase will form. Okay, this active kinase will activate the enzyme by phosphorylation. Once enzyme is activated via phosphorylation by these kinases, then biological effect, that enzyme, the biological effect will occur. That means that enzyme is ready for catalytic activity, its own catalytic activity. That's all. Now selected list of enzymes activated by calcium and mediated by calmodulin. Okay, activated by calcium, which is mediated by calmodulin, calcium calmodulin complex via the protein kinase, right? Adenyl cyclase, calcium dependent protein kinases. This one you have to remember. Adenyl cyclase, calcium dependent protein kinases, calcium magnesium ATPase, glycerol 3-phosphate dehydrogenase, glycogen synthase, glycogen synthase. Glycogen synthase, it is the rate limiting or key enzyme for what metabolic pathway? What metabolic pathway? Glycogen synthase. It is a key enzyme.
Gluconeogenesis.
Gluconeogenesis. What is gluconeogenesis? Gluconeogenesis is the production of the carbohydrate from the non-carbohydrate source, like
Glycogenesis.
glycogenesis. Glycogenesis means glycogen synthesis. Glycogen synthesis enzyme itself, it signifies this one, no, metabolic process. Glycogen synthase means synthesis of the glycogen, isn't it? Glycogenesis, isn't it? Okay, but it cannot synthesize the glycogen in de novo. Remember, glycogen synthase always required one glycogen primer, that is glycogenin. Okay, it will be asked in your exam. Okay, glycogen synthase cannot synthesize glycogen molecule in de novo. Okay, it needs a primer. Okay, primer. Okay, oligosaccharide chain, that is the glycogenin. Okay, glycogenin primer. In that glycogenin primer, it can glycosylates the glucose moiety. Okay, that one you have to study. Each and every metabolic processes you have to study. Okay, before uttering something you have to think, no, what is this metabolism actually. Okay, sometimes, and this one enzymes name also itself, it indicates the metabolic processes, like glycogen synthase means some is going to be produced, synthesis, glycogen. Okay, glycogen is going to be produced, so glycogenesis, simple. Myosin kinase, phospholipase C, phosphorylase kinase, pyruvate carboxylase. Yes, pyruvate carboxylase, it is an enzyme for gluconeogenesis, isn't it? Gluconeogenesis is the first key enzyme, okay, responsible for first rate limiting or key step. Pyruvate carboxylase, it is an enzyme for gluconeogenesis or neoglucogenesis. Okay, pyruvate dehydrogenase. Pyruvate dehydrogenase, it is a different. Pyruvate dehydrogenase, it is a multi-enzyme complex. Okay. Then pyruvate kinase. Pyruvate kinase, it is an enzyme for glycolysis, isn't it? Glycolysis is the step for substrate level phosphorylation. One of the step for substrate level phosphorylation. Calcium in blood, normal blood calcium is 9 to, this is total calcium. Okay, this is the total calcium, not ionized, total calcium. Normal blood calcium, okay, blood total calcium, okay, normal blood total calcium level is 9 to 11 milligram per dl. Okay, this is the total calcium, not ionized. Okay, total calcium, remember, 10 milligram per dl of calcium ion equal to 5 milligram milli equivalent per liter. Okay, 5 milli equivalent per liter. About 5 milligram per dl calcium is in ionized form and is metabolically active. Okay, only ionized form is, I already mentioned, this metabolically active. Another 1 mg per dl is complex with phosphate, bicarbonate, citrate. The two forms are diffusible. These two forms are diffusible from blood to tissues. Okay, these two form, ionized calcium and this one. Whatever fraction is combined with these negative ions. Okay, these are diffusible. About 4 milligram per dl calcium is bound to protein in the blood and is non-diffusible. Homeostasis of blood calcium level. Homeostasis. Homeostasis of the blood calcium level. There are effective control to maintain this narrow range of blood calcium, that is 9 to 11. Body has some effective control mechanism to maintain this narrow range of blood calcium level, that is 9 to 11 milligram per deciliter. Now vitamin D. Okay, what are the different control mechanism? Via the vitamin D, via the parathyroid hormone, etc., etc. Okay, vitamin D. Cholecalciferol is synthesized from 7-dehydrocholesterol in skin under the influence of sunlight. Okay, this is then hydroxylated at 25th position in the liver and further hydroxylated in the first position in the kidney. Okay, ultimately active form of the vitamin D, that is calcitriol dihydroxycholecalciferol, is formed. Okay, dihydroxycholecalciferol, that is calcitriol, is going to be formed, active form of vitamin D, that means. Then vitamin D and absorption of calcium. How vitamin D is related to the absorption of the calcium? Calcitriol, that is the active form of vitamin D, promotes the absorption of calcium and phosphate from the intestine, isn't it? Calcitriol enters the intestinal cell and binds to the cytoplasmic receptor. Okay, this hormone receptor, calcitriol acts as a hormone, active form of the vitamin D, that is the D3. Calcitriol acts as a hormone. So once it is entered in the cell and binds to the cytoplasmic receptor, that hormone receptor complex will form. Okay, this hormone receptor complex interact with the particular sequence of the DNA, okay, particular sequence of the DNA, and causes this hormone receptor complex entered inside the nucleus. Remember, there are specific receptors or transporter is the nuclear receptor is there to receive the hormone receptor complex, isn't it? That ultimately act upon the specific sequence of the DNA and causes de-repression. De-repression, that means depression, reduction, decrease. Okay, and consequence transcription of specific gene, de-repression and consequence transcription of specific gene, then code calbindin, isn't it? Calbindin. Due to increased availability of the calcium binding protein, absorption of the calcium is increased. It causes de-repression. De-repression. Repression means reduction. Okay, depression. Or de-repression means release of this repression. Okay, release of this inhibitory mechanism. That means indirectly activation, isn't it? Induction. Repression means depression. Okay, reduce the activity. De-repression means this reduced activity is released. That means indirectly activity is enhanced. Okay, induce. Okay, it induce the gene that is responsible for coding calbindin. Okay, this calbindin causes increased calcium absorption. Okay, and calbindin synthesis will be more, and this calbindin, it is a calcium binding protein and calcium absorption is increased, like that. Very easy. Vitamin D and bone. Vitamin D is acting independently on bone. It increases the number and activity of the osteoblast. Okay, bone forming cell, osteoblast. Okay, it is called deposition of the bone forming cell, isn't it? Calcitriol stimulate the osteoblast to secrete alkaline phosphatase. Okay, once the activity of the osteoblast is increased, it secretes the alkaline phosphatase. Okay, the enzyme alkaline phosphatase. Due to this enzyme, the local concentration of the phosphate is increased. Okay, alkaline phosphatase, due to this activity of the alkaline phosphatase, the concentration, it causes the lysis of the inorganic phosphatase, both the inorganic phosphatase lysis. So that's why the phosphate ion is released, so concentration of the phosphate is increased. The ionic product of calcium and phosphorus increases, leading to mineralization of the bone. Okay, the ionic product of the calcium and phosphorus increases, okay, which leads to mineralization of the bone. Okay, that is the mechanism. Then vitamin D and renal tubule. Okay, calcitriol, again active form of the vitamin D. Calcitriol increases the reabsorption of the calcium and phosphorus by renal tubule. Okay, therefore both minerals are conserved. Okay, both mineral are conserved. But parathyroid hormone conserve only calcium. You have to remember calcitonin and calcitriol are different. Okay, don't confuse calcitonin along with the calcitriol. Okay, it will be a very big mistake. It will be a blunder only if you confuse the calcitriol with calcitonin. Okay, but calcitonin is a different hormone, isn't it? Calcitonin is synthesized by the parafollicular cell of the thyroid gland. Okay, parafollicular cell of the thyroid gland. Okay, it decreases the blood calcium level. Okay, it is responsible for depletion of the calcium. Okay, it decreases the blood calcium level. L-calcitriol is the active form of vitamin D. It increases the blood calcium. Why vitamin D active form is calcitriol, but chemically it is 1,25 dihydroxycholecalciferol, right? Anybody can tell me? Huh? Please go and check and tell me, because cholesterol metabolism also finish, cholesterol structure also finish. Okay, what are the substances synthesized from the cholesterol also done, isn't it? Structure of the cholesterol also finished, no? So from there only you can tell me, trace and tell me why vitamin D is known as calcitriol. Okay, calcitriol, because the name is 1,25 dihydroxycholecalciferol, dihydroxy. Okay, why it is calcitriol? Tell me in the next class. This calcium homeostasis, when calcium is low, parathyroid hormone is stimulated, resulting in increased calcium release from the bone and decreased renal calcium excretion. Parathyroid hormone also stimulates the increased production of calcitriol, which acts to increase absorption of calcium from intestine. What is parathyroid hormone? It is a 35 amino acid peptide. It is biologically active. Control of release of hormone by negative feedback by the ionized calcium in serum. The release of hormone is mediated by cyclic AMP. Okay, cyclic AMP. The normal PTH level in serum is 10 to 60 nanogram per liter. Nanogram per liter. Very less. In primary hyperparathyroidism, this is increased up to 100 nanogram per liter. Mechanism of action of parathyroid hormone. With this slide, I will finish. The rest of the 2-3 slide I will discuss in the next class along with the phosphorus metabolism. Mechanism of action of parathyroid hormone. PTH activates the adenylyl cyclase, which consequent increase in intracellular calcium concentration. Okay, a kinase is activated and enzyme systems are activated. Okay, PTH and bone. In the bone, PTH causes demineralization. Okay, remember demineralization. The number of osteoclasts also increase. Okay, vitamin D is responsible for increased osteoblastic activity, but here in the PTH it is responsible for increased osteoclastic activity. Okay, as the number of the osteoclasts is increased, the its activity of the osteoclasts also increase. Osteoclasts release lactate. Okay, which solubilize the calcium. Okay, osteoclasts release the lactate which solubilize calcium. In other hand, the osteoblasts release the alkaline phosphatase, which causes lysis of the inorganic phosphate and increase the availability of the phosphorus and causing demineralization, isn't it? Deposition of the deposition of the mineral in the bone. But here in the osteoclast release the lactate. This lactate is responsible for solubilize the calcium. Okay, by this it causes demineralization. PTH and kidney. In the kidney, PTH causes decreased renal excretion of the calcium and increased excretion of the phosphate. Parathyroid hormone action in intestine. It stimulates the alpha hydroxylation of 25 hydroxycholecalciferol in the kidney to produce calcitriol. Okay, to calcitriol. This indirectly increases the calcium absorption. Okay, in the kidney, hydroxylation process of the 25 hydroxycholecalciferol is stimulated by PTH. Okay, PTH. Rest of the slide I will discuss in the next class. Okay.